on the basis of X-ray crystal structures, we can see what's going on. How does one entity, such as an antibody, recognize another? For example, another protein molecule. Well, at that binding or interaction surface, we see a number of individually weak interactions. These are electrostatic, plus minus, hydrophobic interaction, on the basis of X-ray crystal structures, we can see what's going on. How does one entity, such as an antibody, recognize another? For example, another protein molecule or bond. Well, at that binding or interaction surface, we see a number of individually weak interactions. These are side chains, electrostatic, individual amino acids that are at the surface. These side chains can be polar and nucleophilic, they can be hydrophobic, they can be aromatic, acidic, or basic. And it is these individual weak interactions, complementary interactions, that result in a tight fit and binding. And so here again, a few examples of an X-ray structure of an antibody antigen. The antigen is what the antibody binds to. In this particular case, the antigen is lysozyme, a protein, and in the yellow and blue colored regions, that's the antibody. And at that interface, okay, there are perhaps 20 to 30 amino acid side chain contacts and these are complementary. And in a little bit more detail, here's a similar on an X-ray crystal structure. The highlighted area, area that I've shown with a yellow are individual amino acid side chains interacting between the two biologicals, the antibody and the antigen complex. Okay? So this is how molecular recognition takes place at the molecular level between an antibody and its antigen. It involves weak intermolecular interactions for protein-protein interaction. Okay? So, antibodies must seem like the perfect solution. And in fact, they are the gold standard. Do they have any disadvantages? Well, sure. Nothing is perfect. First of all, that's and sometimes it's very, very far from perfect, and so there are some downsides or problems with antibodies. First of all, they are very complicated protein molecules. Their synthesis and purification, okay, can be very challenging. And as I mentioned, the purification alone adds more than 50% to the cost of these. Since they're produced in living organisms, they're susceptible to biological contamination. The production of an antibody from start to finish is timely. Okay? Probably one of the more important factors is the cost of the operation to produce an antibody. And finally, antibodies are proteins. They have a small window in which they're stable and are able to perform. So their stability creates some issues for applications. Okay? And this is where we come in. We are asked the question several years ago, can synthetic polymer nanoparticles be engineered with affinity and selectivity for biological macromolecules? That is to say, can we make something in a chemistry laboratory inexpensively and quickly that might actually function as an antibody? So I've put a few slides in, and I'm being a little bit more careful this time about our time. And so I'm going to be skipping around a bit or running my way through certain slides. And I'm going to jump to this slide, which gives us a basic concept of how we think about making something in the chemistry laboratory that can function like a like an antibody. Once again, an antibody, let's say protein A, and its target, protein B, involves the collective interaction of very weak interactions between side chains of amino acids. If we were to produce something in the chemistry laboratory, what we would need is a synthetic polymer nanoparticle that had complementary functional groups to the surface of the protein that we'd like to bind to. Now, here's the problem. Our synthetic polymer nanoparticle, as I'll mention shortly, is made by a polymerization reaction. We cannot control how these side chains are arranged in three-dimensional space. What we're going to be making is a random polymer. And so what we do in order to compensate for that randomness is that we make our polymer nanoparticle very flexible. And hopefully the functional groups on the polymer nanoparticle can find their complement, and the polymer nanoparticle adapts to the surface of the protein target to give some level of affinity. Okay? I'm going to quickly go through an example which sets the stage. In this particular case, our initial target was a peptide, melittin. It's a cytotoxic peptide that's the principal component of honeybee venom. This particular peptide is comprised of hydrophobic brown residues, positively charged blue residues, and hydrogen bonding amino acids, green. We're going to be making polymer nanoparticles, nanogels or microgels. There's sometimes confusion used in calling these, but we're going to be doing a polymerization reaction in water from readily available monomers. The picture that I have on the slide at the top, there's one monomer that has an X group. That X group will incorporate functional groups, hydrophobic or charged or polar, and that will be incorporated into our nanoparticle. The polymerization reaction occurs in several hours at 70 degrees, and at the end we have a very uniform solution of polymer nanoparticles. Some information about what we control is below. The polymerization reaction gives a random copolymer. That is, the functional groups are randomly distributed. And now we have to address what seems to be a conflict with the central dogma of molecular biology. That is, in molecular biology, there is a residue-by-residue transfer of sequential information. And so the central dogma consists of the fact that biological information is encoded in linear sequences. Yes? In what? Yes. Well, certainly the biological information in DNA and in RNA and in proteins, we are now at the nanoscale. The point here is that if we think about the concept of information in DNA, RNA, and in proteins, it is encoded in the linear sequence. And maybe this is a little clearer on this and the following slide. So we're talking about the information contained in DNA. It is the specific sequence of nucleic acids in DNA that is the source of the information. That information is transcribed into RNA and then translated into a specific protein. Yes. Yes. That is, with CRISPR technology, that is profound. And I think the entire world should be very sensitive to the potential consequences of that. After learning that this biological information, this is the blueprint for you and me and everyone else. It's contained in our DNA, okay? We now are beginning to enter an era when we can edit that information. And it is technology, and I think whether it's good or bad depends upon how it's used, obviously. There is a potential to correct and edit, for example, certain genetic diseases. That possibility exists and will continue to be refined as we understand CRISPR technology more and more. But I think everyone could understand that the potential is enormously important, and we must proceed extremely cautiously. Yes. Yes. That was extraordinarily irresponsible. And I think there should be penalties and consequences for that. Yes. Yes, that would be correct. But what is frightening is that there are private companies that would have access to that technology. And so the consequences of this are extraordinarily profound, and we must proceed very, very cautiously. Okay? Oh, I certainly hope not. That would be, again, extraordinarily irresponsible. As I said, this requires the attention of not only the scientific community, but the broader community of the world to stand in judgment about what might be done, or what could be done, or what should be done with this information. And the consequences, these are blueprints for you and I. And the concept that someone could tinker with that and alter it, one can, as I said, perceive potential profoundly important and useful consequences in very carefully controlled, selective situations. But on the other side, one can imagine just a horrible misuse of this technology for self-interest or goodness knows what. And so this is a point in history that I think we must mark and pay very, very careful attention to and insist upon a much broader discussion of any applications of this technology. Mm-hmm. Yes. Well, I wouldn't call it a paradise. I would, yeah. I guess all I can say is that one must be very, very cautious about considering any practical application of this technology, which is, in my opinion, is far from perfected. The consequences of this are not yet appreciated, nor has the techniques been perfected. So, again, I'm just as a concerned citizen with some scientific literacy want to exercise extreme caution in this area, because we've entered a new era in terms of the human race. Yes, absolutely. Right, right, exactly. And quite frankly, it may be even more consequential for the human race than nuclear energy. Right. Okay, so once again, I'm going to have to skip. So the central dogma of biology is that information is encoded in linear sequences, DNA sequences, RNA, and then finally the information in the sequences of amino acids in the peptide. That information encodes function. We're going to be dealing with synthetic polymer nanoparticles that don't have encoded linear sequences, but random sequences. And so the big question, can they ever be produced in a way to have function similar to, for example, antibodies, proteins? I'm going to go back to how we make our synthetic polymer nanoparticles. These are polymerization reactions which result in the formation of very uniform nanoparticles. These polymerization reactions are run in water with functional monomers that impart charge, hydrogen bonding, hydrophobic interactions. And we make combinations of those functional monomers in the polymerization reaction to produce a library of nanoparticles of different composition, which we then evaluate in the chemistry laboratory for affinity to a particular molecule. In this particular case, it's the peptide melatonin. And we discover that there are a number of, there are several nanoparticles in our library that bind melatonin to the peptide. And these are our lead candidates. We then go and perform other diagnostic tests to confirm that our nanoparticles, or selected nanoparticles, bind and the peptide melatonin. And here we use a red blood cell test. Melatonin breaks apart red blood cells. And in the first test tube, we have red blood cells that have been centrifuged. The second, we've added melatonin into red blood cells and then centrifuged. And we notice that the supernatant is red. That's because the red blood cells have been broken open by the toxin. and hemoglobin, a small red protein, stays in solution. Subsequent test tubes are red blood cells, melatonin, plus our nanoparticles. The important point is nanoparticle 6 seems to bind to and inhibit melatonin. And this is how we discover synthetic polymer nanoparticles that bind to, with selectivity, to the target biomacromolecule. Okay? We have taken these nanoparticles, once we have refined them, and demonstrated that they can actually neutralize the toxin in the bloodstream of a living mouse. And these experiments confirm that our synthetic polymer nanoparticles, produced rapidly and inexpensively in the chemistry laboratory, can actually function in a very complex environment, such as the bloodstream of mice that have been inoculated with a deadly toxin. Now I'm going to jump forward. The lessons learned from these preliminary studies is that we can make polymer nanoparticles with affinity to protein targets. They can be optimized by a process that we call directed chemical evolution, by making incremental, systematic changes in the chemical composition of the nanoparticle. And I'll just illustrate that with the example that I've chosen or discussed previously, melatonin. Here we're looking at the lower right at the performance of our nanoparticle, a collection of nanoparticles where we systematically vary the percentage of the two functional monomers shown on the X and Y axes. And the neutralization is the ability of that particular nanoparticle to capture, sequester, and neutralize melatonin. And you can see that the performance is very sensitive to the relative amounts of these two functional monomers shown on the Y and X axes. Okay? And we're able to produce a nanoparticle with nanomolar affinity. We have shown this in a number of examples, including more complicated protein captures. Okay, there's not time to go into the details, but this is work that has been published from our laboratory over the last 4 or 5 years. I'm looking at the watch, and I promised I would get to the important topic of snake envenomation. So I'm going to summarize what we have learned so far with the one example that I showed in some detail. We demonstrated that it is possible in the chemistry laboratory, and I'm going to go back, in the chemistry laboratory to synthesize a polymer nanoparticle with functional groups, and we can, by adjusting the relative amounts of these functional monomers, we can design nanoparticles with very high affinity and selectivity to specific protein targets. That's the basic important lesson that we have learned so far. It seems to be in conflict with this central dogma of biology that information is contained in the linear sequences. Our materials are I'm zipping through are random polymers, and so from an information content it has very, very little information, and yet we can achieve many of the functions of an antibody, at least in selected cases. We just recently acquired an image of what our nanoparticle that has captured a protein looks like, and this is a cryo-TEM image. It's a little bit fuzzy. Our nanoparticle is very low density, The dark spots on that central feature are proteins that are embedded in this nanoparticle. Okay. So we have our very first images. The nanoparticle itself is 20 nanometers in diameter. Okay, that's just something that we achieved recently. And now I'm going to get to this topic of using our technology to produce a nanoparticle that can bind a protein or a peptide. We're looking now at what I think are very important applications. This problem was selected for several reasons. One is, as we'll see, snake envenomation is a very important problem worldwide. It is a problem that has no simple solutions. That is to say, there are antibodies that work against snake venom. But those antibodies suffer from some of the limitations. And in this particular problem of neutralizing snake venom, there are special complications that make traditional antibody solutions less than perfect. So our goal is to develop a synthetic polymer nanoparticle or nanoparticles that are capable of sequestering and neutralizing the important protein toxins that are found in snake venom. We have identified at least 3 main protein toxin families that are found across all species of venomous snakes. These are phospholipase, 3-finger toxins, and the metalloproteinases. Those 3 protein toxins in particular contribute importantly to tissue necrosis. These are the terrible disfiguring consequences of snake envenomation that oftentimes result in the loss of a limb. Yes. Yes, well, that's an important point, and I'll get to that, in fact, on the very next, on the following slide. The goal is to provide coverage across all species of venomous snakes. And I'll show in a moment why this is a particularly challenging problem, because snake venom, as we'll see, okay, varies significantly from species to species. And this is one of the complicating factors for traditional antibody antivenoms. First, the worldwide problem of snake envenomation. It's been elevated by the World Health Organization to a category A, neglected tropical diseases. There are more than 100,000 deaths per year resulting from snake envenomation. And 750,000 to 1 million people suffer serious morbid injuries. The map, which was put together by the World Health Organization, identifies regions in the world, primarily along the tropics, that are most affected by snake envenomation. And we see Brazil, of course, is prominent in South America. Also, Sub-Saharan Africa, India, and Southeast Asia, okay, are particularly afflicted. I've put several species of snakes that might be familiar to Brazilians. Hopefully, none in the audience have encountered these, because I think, as you all know, these are venomous snakes that can be commonly found in Brazil. Okay. Yes. It may not be a good friend to many people, but I'm sure people are aware of it. Right? And so we ask the question, what is snake venom? And what makes the venom such an important contributor to world health? Well, it turns out snake venom is a complex mixture of protein toxins. It's just not a pure substance. They are optimized for the snake to immobilize or kill its prey and actually to initiate the digestion. Protein toxins, as I indicated, can be fit into broad subsets of functional protein families, including PLA2. That's an enzyme that breaks down cell walls and membranes. Three-finger toxins, which are cardiotoxins and also neurotoxins and cytotoxins. Metalloproteinases, those digest proteins, okay? Protein inhibitors. The relative amount of these protein families is species specific. This western dimelback is something that I can sometimes find in around my home in Southern California. As I indicated, the composition of venom is species specific. And so the many branded crate consists primarily of three-finger toxins and phospholipases, in addition to several other types. On the other side, in the viper family, the western diamondback rattlesnake, its venom consists primarily of metalloproteinases, okay, and serine proteinases. These are enzymes that break down proteins, okay? And so each snake species has its unique cocktail of venomous proteins that complicates the development of a broad spectrum antivenom that would be effective across all species of snakes. The problem is even more complicated in that these protein toxins are not pure substances. Perhaps in an effort for the snake to stay ahead on an evolutionary basis of its prey, oftentimes small mammals, the protein toxins are not pure substances, but consist of many isoforms. These are taking the coded information in DNA, but putting those pieces together differently. And so each protein toxin, for example, PLA2, consists of a collection of isoforms. And so, for example, in the three species of snakes that I've indicated here, there are over 135 different PLA2 isoforms. And so this, of course, raises the ante, or makes it more complicated to produce a broad spectrum antivenom to sequester entire families. Traditional antivenom, some of which is produced, of course, in Brazil, is done by collecting the venom, inoculating of a specific species of snake, inoculating a large mammal, typically a horse or a goat or a sheep. And yes, a horse, yeah. Careful as to how much venom is inoculated. It's done multiple times. One doesn't want to injure the horse, hopefully. But then the blood is drawn, and then an extensive purification process to isolate the antibodies from the horse, only some of which are antibodies that are produced in response to the venom that was introduced to the horse. Many of the antibodies have nothing to do. And so these are not human antibodies, they are equine antibodies, and they run the risk of individuals having an allergic response. The antibody or the antivenom itself must be introduced in a clinical setting by an IV drip. And now you can understand the importance of identifying and having on hand the antivenom for the specific species of snake responsible for the bite. Sometimes that information or that antidote is not available. Okay. So what we set out to do in the remaining slides, and I hopefully will finish on time, is to identify the three main families of protein toxins that are found across all species of snake. PLA2 is one of them. PLA2 is an enzyme that breaks down cell walls. That's the start of a process that results in this terrible digestion of tissue that's responsible for these morbid injuries. And so in a chemistry laboratory, we make libraries of polymer nanoparticles containing different organic functional groups. And then we develop an assay that allows us to screen whether any of the nanoparticles in the library that I've shown on this slide can effectively sequester PLA2. And this is a collection of isoforms in a specific venom. And here we show our first hit, which is a nanoparticle with the chemical composition shown at the bottom that seems to have efficacy for binding PLA2, that enzyme, from venom. We then go on to examine whether that nanoparticle candidate is capable of actually not only binding the protein toxin, PLA2, but also of inhibiting its function, preventing it from breaking down cell walls. And on the basis of this screen, we have identified and improved the formulation so that we have a nanoparticle that not only binds to PLA2, the green molecule, the protein, but also inhibits its function from breaking down cell membranes. Okay? And this is the nanoparticle that we finally have come up with. Now what's important is to test the nanoparticle in more challenging situations using whole venom and in the presence of serum proteins. Because if we had a nanoparticle that bound all proteins, it wouldn't be selective. And so we have to demonstrate selectivity. And so this is a chromatography experiment, an STS page, okay, that shows on the first column that I've highlighted just the venom itself, PLA2. Column number 3 is all of the proteins in serum. Now, if we mix our nanoparticle with just serum and isolate or identify what the nanoparticle picks up, we see it picks up several proteins from serum, showing that it's not particularly selective. But if we mix venom plus serum and then nanoparticle, we show that, in fact, that our nanoparticle is able to selectively pick out of, from a mixture of venom and serum proteins, the toxic protein. Okay? This is a protein from the spitting cobra. Another example we show in a similar series of experiments where we expose our nanoparticle to serum proteins, more than 2,000, and the venom. The nanoparticle is able to, sorry for jumping around, selectively pick out the venom proteins which are circled in the white box, okay, on this particular slide. So the nanoparticle has been shown that it can selectively capture venom proteins in the presence of many, many thousands of different proteins. And we go further using proteomics to identify the proteins that the nanoparticle picked out, and that's to make sure that the nanoparticle picks out proteins that are associated with the snake venom. Use a technique called proteomic analysis, which unambiguously identifies that the nanoparticle is selectively sequestering and removing from this mix venomous proteins. And in this particular case, we see this one nanoparticle actually removes two toxic proteins, PLA2, three-finger toxins, in their isoforms. We have some chemical explanation for why our nanoparticle is able to function in this capacity. It's not particularly important, but suffice to say that the nanoparticle will seem to mimic the natural substrate of these two families of venomous protein toxins, PLA2 and three-finger toxins. Okay? In our just to finish, I have one last slide, unless you have a question. Okay, this is the last slide. In a collaboration with Professor Gutierrez, okay, University of Costa Rica, I'll have a picture of him in a moment, we have been able to demonstrate that this single nanoparticle is able to show efficacy in vivo, that is, in mouse models. And so this is an assessment of the tissue necrosis that's responsible by injecting in the skin of a mouse some of the venom, okay, from a specific snake venom in this particular case. On the far right is the extent of tissue necrosis found by just injecting the nanoparticle. We have to be certain the nanoparticle itself is not toxin. But in this particular case, if we mix nanoparticle and venom and inject, or inject venom with a follow-on injection of nanoparticle, we see a significant reduction in the extent of tissue necrosis. Okay, and so this is the first very exciting and very promising result, indicating that perhaps we now have an opportunity to produce synthetic polymer nanoparticles inexpensively. These are non-biologicals that we might be able to incorporate in an injectable device that could be carried by people in the field. And in the unfortunate event of envenomation, they would be able to immediately inject at the site of envenomation this antidote and suppress these fast-acting enzymes that break down tissue and result in these horrible disfiguring effects of snake envenomation. So we have established now a working collaboration with Professor Gutierrez, okay, who is a world authority on snake envenomation, and his institute in Costa Rica is responsible for producing, okay, a good deal of the venom that's used in Central and some in South America, as well as Sub-Saharan Africa. And so we are undergoing more extensive tests to hopefully validate the use of synthetic polymer nanoparticles, okay, in our effort to produce a truly broad spectrum antivenom. Now, I'm a professor and I don't do any work. All of this is done with my students and postdoctorals at the University of California and in collaborations, as I've indicated, in Costa Rica and elsewhere. And I'm indebted to them. And then finally a picture of the students. That's me in the middle of the table, left the old man. And these are the young students whose ideas and hard work have resulted in the developments that I talked to you about. Hopefully I'm not too far over time as I was last time, but I did get yes. Yes. I'm very grateful for that, and I am indeed very interested. It was my intention to, after this presentation, to get back to you with help in establishing contacts and connections. As I said, interestingly, snake envenomation is not a particularly important problem in the United States. And so finding collaborators and expertise and people with genuine practical experience is a little bit more challenging. But as I reach out to countries where this is a very serious and real problem and where experts reside, as I said, I'm a chemist. I don't have specific expertise in snake envenomation. Professor Gutierrez does. But I would like to reach out to as many other locations, people with expertise that we can draw upon in our effort to move forward in producing something that is relatively inexpensive and readily available that could perhaps save lives, but certainly prevent the terrible disfiguring consequences resulting from snake envenomation. We think we can have an impact in this area, but it will require collaboration with groups with expertise and practical experience in these efforts. So it was my intention to follow up with your generous offer to establish connections between these groups in Brazil. Yes. Yes. Very good. Thank you. It was a pleasure and an honor to participate in your program. Bye-bye.